Hi everyone, welcome back to Useful Genetics. This is Lecture 1K, which is going to be a very short overview of the marvels of modern DNA sequencing. So we'll talk about what the technology differs, how it differs from what we used to be able to do, and the different things that we can learn using this technology. So back in the good old days, before about the year 2000, DNA sequencing was done on pure samples of DNA consisting of many identical copies of a single DNA molecule, single DNA fragment, usually. And the result of that sequence was, of that analysis, was one DNA sequence that represented the average sequence over the whole sample. Modern DNA sequencing, in contrast, can take a sample that consists of a mixture of many different DNA molecules, and it can sequence each molecule in the sample, giving many different sequences as what are called sequence reads. Modern DNA sequencing is also an awful lot cheaper than it used to be. Here's a graph showing the cost of DNA sequencing as it's changed between the year 2001 and the year 2013. And it's compared with the Moore's law decrease in the cost of computer power, which goes down. I think Moore's law has it that the cost of computer power falls by half every 6 months. So for the first 6 or 7 years, the cost of DNA sequencing decreased dramatically, roughly at the same rate as the cost of computing power. And then, starting in about 2007, it plummeted. And now it's back to still decreasing dramatically, but only at about the rate of Moore's law. So DNA sequencing has become very cheap. Well, cheap for science. Nothing in science is really cheap. Now, I'm going to describe 3 applications. And the first is sequencing not just a gene, but a whole genome. So for an organism with a relatively small genome, we can take a single sample tube with the entire genome broken into fragments and sequence all the fragments. And then by comparing the fragments, we can line them up and find the overlaps that allow us to infer the complete genome sequence from the assembly of these short reads. For a larger genome, we can do the same thing, but we'll need to use multiple samples. Now, a second application is that rather than sequencing one genome, we can sequence a mixture of DNAs from different sources. DNAs from all the members of a population or from all the species in a community. For instance, you can scrape the bacteria off your teeth and extract the DNA from this mixture of bacteria and sequence it. This is a study called metagenomics, studying the phenomenon of the human, what's called our microbiome, the ecological communities of bacteria that live on us. And when these sequences are analyzed, they can be grouped into particular groups that tells you the kinds of organisms that are present in this sample, even though you don't have any way to directly study these organisms. The third application of DNA sequencing is measuring RNA abundances in a cell or tissue. We've known for many years that different genes are expressed at different levels, and there's different amounts of RNA for different genes in the cell, but we didn't have an easy way to measure this. We still don't have a way to sequence RNA, but we do have a very easy way to use an enzyme to make DNA copies of the RNA, and then we use our very efficient DNA sequencing method to sequence all of the complementary DNA, it's called, in the sample, which gives us a measure of the sequences of the RNAs in the original sample. We can then basically count the amount of RNA sequence that we get for each gene in the genome, and that tells us how much RNA is present in that cell or tissue from each gene. So this is a gene that's expressed at a very high level. This is a gene that's hardly expressed at all. So what we've done, we've talked about how the new methods of DNA sequencing can sequence many DNAs at once rather than just a single molecule, and they can do it far less expensively than the old methods did. This enables very efficient DNA sequencing to be applied to new problems. We can sequence whole genomes much more easily, for instance, than we sequenced the human genome when we started. We can characterize the genetic membership of ecological communities just by sequencing the DNA without actually knowing what the organisms are. And we can use DNA sequencing to measure the abundance of RNA in a sample, which tells us how strongly different genes are expressed in different tissues. Coming up next, we're going to switch to think about an evolutionary concept, the concept of homology, which is vitally important for many aspects of understanding genetics. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is Lecture 1L. We're going to talk about the very important concept of homology. We'll talk about its meaning in biology, and we'll particularly apply it to genetics. We'll talk about how to decide if similarities are due to homology or due to other factors, and we'll talk about the specific genetic case of homologous chromosomes. Now, I'll start with a little justifications for why we talk about evolution a lot in a genetics course. The quote is from a noted geneticist, Theodosius Dobzhansky. "Nothing in biology makes sense except in the light of evolution." This is particularly true for genetics. Heritable genetic variation is what genetics is about, but it's also what evolution is about, since heritable genetic variation is what makes evolution possible, allows natural selection, and it's what natural selection acts on, and it's what has evolved. So all of the features of the genetic systems that we study are the products of evolution. So we're very much embedded within an evolutionary world, as is all biology. Now, homology is often confused with similarity, and that's because homology is a special kind of similarity. Lots of things are similar. Things that are homologous are similar because of shared ancestry. So here are examples of the four-limb bones of four different vertebrate species, starting with the human arm bone. And they've been colored to show up their similarities. So there's the upper arm bone is a big bone. Then the lower arm, our forearm, has two bones. Then there are the small bones of the wrist and the long bones of our fingers. And we see similar arrangements of bones in the forelimbs of all other vertebrates. And this is because the ancestor of all these vertebrates had a limb with this structure. This is called the tetrapod limb. So it's shared ancestry that has given us these similar features. Shared ancestry also applies to the genes that are responsible for the development of these features. For instance, here we're comparing the genes that control embryonic development in a fruit fly and in a mouse. And the genes are colored according to their function and positioned according to their arrangement on the chromosome. And we see that the genes are similarly colored for similar parts of the embryo, and they're arranged similarly on the chromosomes. They also, although you don't see that here, they have similar sequences. And we now know that genes like this control embryonic development in all animals, and that these genes are similar because they are descended from a common ancestor of all animals that controlled its development with these genes. We can extend the comparison farther to look at individual sequences. Here are the amino acid sequences of two genes. One gene is the eyeless gene of fruit flies. When this gene is defective, the fly doesn't have any eyes. The other is a gene that was studied completely independently in humans. It's responsible for a disease called aniridia, which is the hereditary absence of eyes. once these genes were sequenced and compared, it was astonishing to see how similar these sequences are. The red amino acids are the ones that are different. Almost everything else about these sequences is identical. And this level of similarity must be due to common ancestry. And this tells us that the ability to develop an eye, although the types of eyes are very different in fruit flies and humans, that the ability to develop an eye is an ancestral feature that was controlled by this gene in the common ancestor of all animals, particularly humans and fruit flies. Now, not all similarity is due to homology. That's part of why there's this confusion. Similarity can simply be due to chance. This is especially true for relatively simple features. For example, if we're comparing just short segments of two DNA sequences, we'll often find short strings of bases that are the same simply by chance, with no evolution or ancestry involved. Similarity can also arise on independent features that become similar because natural selection is selecting for the same function. Here we see two animals. This is the Australian echidna. Its only close relative is the platypus. It lays eggs. This is the mammal, European mammal, the hedgehog. Both of these animals eat insects, and both of them have transformed their fur coats into spines. We know that this is not homologous because we know a great deal about the ancestors of echidnas and the ancestors of hedgehogs, and they don't have spines. They have independently been selected for spiny coats as a form of defense. Finally, similarity can be due to common ancestry, the kind of homology that I described in the previous slide. Now, how do we decide whether similarity is really due to shared ancestry? The answer is it's not always obvious, but usually we have a lot of other information. The general principles are if the similarities are so strong that they couldn't have arisen by chance, for instance, the similarity between the aniridia gene and the eyeless gene that I showed you, and they're too arbitrary to have arisen by convergent evolution. That is, the similarities extend to features that would not be acted on by natural selection. Then we infer that these similarities must have arisen because of divergence from a common ancestor, and that is, they must be due to homology. Now, once we've decided that features are homologous, we can use them to tell us more about evolutionary processes. And this is particularly true for DNA sequences. Once we've decided that DNA or protein sequences are homologous, there's a lot of other information in there that lets us make evolutionary inferences, the simplest of which is knowing how similar two homologous sequences are tells us how recent their common ancestor was. So here's homologous sequences from 3 different species. And we see that the first 2 species differ only at 2 positions. So we infer they had a very recent common ancestor. However, sequence 2 and sequence 3 are much more different. They're different there. And we infer that sequence 3 has a more distant ancestor from sequence 2 than sequence 1 does. We'll talk more about this in module 2. Now, in genetics, homology is used in a particular way in one particular case. It's used generally in genetics, but in particular, it's used in the term homologous chromosomes. It's really the same meaning that homologous has everywhere else, similarly due to shared ancestry, but it particularly applies to the 2 versions that we have of each of our chromosomes. So our chromosome 7 for mom and our chromosome 7 for dad are referred to as homologous chromosomes. They are, of course, truly homologous because the reason they're so similar is because they're descended from very recent common ancestors, not species way back in evolutionary time, but humans who were our ancestors maybe only a few thousand years ago. So they're very similar because they share very recent common ancestry, and we refer to the different versions of any particular chromosome that humans have as being homologous chromosomes. Now, here's a question for you. Which of these pairs of entities are homologous? And I'll just point out one thing about this question, which is that the answer boxes are square. Now, you might think, they just picked squares instead of circles. But in fact, in Coursera and in many other multiple choice situations, square answer boxes have a special significance. They mean that this is a question where there may be more than one correct answer, and that you will be able to check more than one box in your answers. Well, I hope that you did check more than one box. I hope you checked all 3 boxes that are entities, not that one, entities that are indeed homologous. The 2 that are not homologous, your chromosome 7 and your chromosome 17, they have completely different genes. They are not homologous at all. And of course, a mouse and a computer mouse, they're named the same because they look similar, but they're completely independent entities. Now, what we've done, we've talked about homology, which is in some ways a very difficult concept, but definition-wise, it's very similar. Similarity that exists because of shared common ancestry. And the trickiness can come in deciding where it applies, but you will only be encountering situations where it's really quite obvious that it applies. And we talked about the specific case in genetics of the different versions of a single chromosome that we have, which are referred to as homologous chromosomes, because that's what they are. Coming up next, we're going to talk about ploidy and sexual life cycles. I hope to see you there.